morning. Thank you to be here. I'm Rosemary. I'm a PhD, next PhD. Today I am going to talk about the construction of EEG-based functional network model based on graph theory, and I will show you the results of a study applying this method to compare the pathophysiological characteristics from various psychiatric spectra. The pipeline of this method starts with EEG recording and preprocessing. Then a source reconstruction is performed to estimate the neural source activity and reconstruct their temporal dynamics, with a source space defined by a region of interest, often with an atlas-based approach, as in this case 68 regions of interest are taken by Desikan Atlas. Then a statistical coupling between the regional time series is computed to estimate the functional connectivity between them, and a functional connectivity matrix is used to study the topological characteristics properties of the network based on graph theory metrics. And this is useful for clinical application because we can explain a complex functional network with a small measure, for example, for localizing epileptic networks in an epileptic patient, or to derive reliable biomarkers of cognitive decline in neurodegenerative disease. But the key step of this pipeline is the transition from the electrode space into the source space that consists of solving an ill-posed inverse problem, that is the determination of the generating sources of the brain's activity. Let's take a step back to what EEG is and what its records. EEG is electroencephalography is a new technique. It has been used from over 80 years to study the normal and pathological brain activity in clinics. Its record differentials in electric potential captured by two electrodes on the scalp following an international standard 1020 system covering the entire head. The main source of the signal are extracellular postsynaptic potential of the pyramidal cells because of their organization in macrocolumn and the synchronization and then summation of their potential. So each electrode record capture the signal to some extent that arising from all the brain sources, depending on the distance between the source and the sensor and on the orientation of the equivalent dipole. But the voltage field gradient, we know, decreases with the square of the distance. So the activity from deep sources are difficult to detect with EEG. It consists of a two-dimensional matrix, time for amplitude, for each channel. And for each time point, it is possible to show the amplitude of the potential by a topographical maps on the scalp. However, each electrode captures a signal propagation due to the volume conduction. And so the maximal activity that each electrode record capture doesn't mean that the source, the generator, is under that electrode. It follows that the statistical coupling measured by two electrodes cannot be interpreted as a direct measure of brain activity of the underlying generators. And this technique is attractive because a high spatiotemporal resolution network can be directly identified in the search space, overcoming the problem of the volume conduction that corrupt the sensor level recordings. And we can use EEG as an electrical search imaging technique by incorporating temporal and spatial components from EEG signal. The aim of this process is to build a realistic head model by segmenting the MRI to estimate the cortical sources. And to do that, we need EEG recording, the 3D electrodes position on the head, and anatomical data that can be the subject MRI or a template. Then a model of the head and a source model. And first of all, we have to co-register the electrode position on anatomical data by identifying the same fiducial points that are nasion, inion, left ear, and right ear. Then a cortical tessellation is performed into thousands of triangular elements, and a head model that takes into account the different geometry and conductivity characteristics of the brain tissues are used to estimate the current dipole, assign each of dipole to each of triangles, and constraining them perpendicularly to the cortical surface. The outcome of the modeling process is an unleaded field matrix, that is a matrix and sensors per n sources that relate the activity of the sources to the sensor data collected during the experiment. And the most common head model used are the boundary element method, the finite element method, and the finite difference method. With the lead field matrix, we can then build a source model that aims to estimate the amplitude of each dipoles, of each of 1,000 dipoles provided by the forward model. But since the electrodes are limited in number respect of the dozen of dipoles, of the current dipole, it's said that it is a nil-posed inverse problem. And to overcome this, for estimation of the sources, an a priori mathematical constraint is required before the process of estimation in a form of covariance matrix of the source that favor the solutions that are of minimum energy and that can be derived directly from the recording. And the most common method, source model used, so are the minimum norm imaging, the beamforming, and the dipole modeling. But recently, a new approach using machine learning algorithms are employed. In particular, the deep recurrent neural network that have the advantage to faster inverse solution that can be useful for future application in clinics for real-time electrical source imaging. This pipeline, this method, was used in a recent study aimed to compare the fault mode network model among major psychiatric disorders. The author hypothesizes that these models might reflect their pathophysiology. And EEG data were extracted from a database of patients diagnosed with major psychiatric disorders by clinical evaluation of experienced psychiatrists. And a total of 441 patients were selected and were excluded from the analysis those having other neurological disorders or other organic brain damage, impairment in sensory or motor function. And a total of 2,050 healthy control were selected based on demographic information, including age, gender, and education. Resting state EEG was recorded for 4 minutes with 62 electrodes. The signals were segmented in 2-second epochs without an overlap, and 45 epochs were randomly selected for each participant. 25 default mode network ROIs were determined based on literature, including cingulate, frontal, occipital, parietal, and temporal cortices, and hippocampus. For the source localization, a brain template and deep brain structure.
There's a question.
Yeah, sorry.
How this default mode network nodes were extracted based on the fMRI data, or there's some atlas based on REST EEG data?
They searched a lot of papers defining this region of interest with that specifying the coordinates of this region.
Yeah, but because usually the default mode network is like is a construct used in resting state fMRI studies. So
yes,
I
I
am
know.
not sure whether these DMN nodes were taken from this, like power or atlas or something like that, or like there's a specific DMN network based on EEG studies also.
I know, but they don't specify this. They say only that check it out for some papers having defined this ROI with their coordinates.
That's from Atlas.
Some atlas.
I don't know.
There is from Atlas.
25 ROIs, yes? ROIs.
Yeah.
ROIs.
To construct the source signal of each ROI, principal component analysis was applied to the source signal. The source signal was then decomposed in 5 frequency bands: theta, alpha 1, alpha 2, beta 1, and beta 2. And the functional connectivity was evaluated by the phase locking value. The phase synchronization method to estimate the functional connectivity in EEG techniques are the most important method, because for 2 reasons. Because EEG is a direct measure of synchronization, neural synchronization, and because oscillation promotes neural communication and regulates it by improving it if oscillations are in phase, or inhibiting it is out of phase. And for each frequency band, a clustering coefficient was chosen as a graph theory metric. Clustering coefficients represent the tendency of a network to form topologically local circuits. It's a main measure of segregation, of nodal segregation in a network. And the global clustering coefficients was evaluated to quantify the overall clustering level of a network, and local clustering coefficients to quantify the degree of nodal clustering within its neighbor nodes. And at a glance, homogeneous clustering tendencies was found in this specific manner. And in particular, an hyperclustering was found in the theta-band for Alzheimer's disease, mild cognitive impairment, schizophrenia, and bipolar disorders. And in beta 2, hyperclustering was found for major depressive disorders, panic disorders, and post-traumatic stress disorders. Instead, an hypo-clustering was shown in alpha-1 band for AD and MCA, and in beta-2 band for schizophrenia and obsessive-compulsive disorder. And when compared to the corresponding healthy control, a global higher clustering was shown for Alzheimer's disease and schizophrenia group. And in particular, in the region of frontotemporal parietal regions for both group. And these regions are linked to working verbal memory. So an higher segregation in this area led to an efficient cognitive function. Moreover, occipital gyrus was shown to be hyperclustered. And this area is involved with visual systems. So a deficit in this area led to psychiatric symptoms such as illusion or hallucination that are typical of this disease. We can note also that Alzheimer's disease have a widespread hyperclustering among other regions, and this could mean a several neurological degeneration. In alpha-1 band, instead, a lower clustering is what's shown for Alzheimer's disease, mild cognitive impairment, and obsessive-compulsive disorders. And for all groups, in the left lingual gyrus, that is linked to encoding and retrieval of visual memory. So also in this case, a cognitive decline is defined by a lower clustering. But in particular, the MCI groups show a lower local clustering in parietal and temporal cortex. Instead, the Alzheimer's disease group in a widespread had comprehending frontotemporal occipital cortex and hippocampus. And this could mean that a progression of neurological degeneration from MCI to AD patients happens. We know that neural degeneration begins in the temporal regions in MCI patients and then gradually spread to other brain regions in Alzheimer's disease. For obsessive-compulsive disorders, another lower local clustering was shown in post-cingulate cortex. And these regions, this could mean an excessive mental load and obsessive rumination that this pathology, that's typical of these pathologies. And in beta-1 and beta-2 band, a lower clustering coefficient was shown for obsessive-compulsive disorders and a higher clustering for major depressive disorders. The beta-2 oscillation reflecting sustained processes of attention, perception, emotion, and working memory. So, in major depressive disorder, an hyperclustering led to the deficit of these processes because the information is segregated in local circuits. To the contrary, in OCD groups, there is less segregation with a spread of information in other brain regions, resulting in iterative, compulsive, and obsessive behavior. For the functional connectivity, the authors show an heterogeneous pattern of lower and higher connectivity. Only in the theta band between the lingual gyrus and the left hippocampus was shown lower connectivity from these two regions that was significant only for schizophrenia, bipolar group, and post-stress traumatic disorders, but also in less measure for panic disorders and major depressive disorders. And also the results might be related to cognitive decline that is well-known pathophysiology of those psychiatric disorders. So the results showed that this method is clinically useful for facilitating the association of analysis between the analysis results and neurobiological characteristics and for deriving reliable biomarkers from various spectrum of the disorders. However, although the authors assume that sub-abnormal feature of the fault mode network may be related to a cognitive function, there is no, in the paper, a cognitive assessment underlying this argument. So future studies should be needed to reveal the relationship between these findings and cognitive function. And thank you for attention.